All of you, Irene Maroto. Irene graduated in biology at the University Complutense of Madrid, and then she obtained a master's degree in neuroscience at the Universidad Autónoma de Madrid. She completed her PhD in 2021 in Professor Manuel Guzmán's lab with an FPU fellowship. She received the Juan Abelló Pascual Award by the Royal Academy of Doctors of Spain. And she also made a couple of pre-doctoral and postdoctoral stays at the Pablo Castillo's lab at the Albert Einstein College of Medicine in New York. And recently, she has obtained a postdoctoral Margarita Salas Fellowship. Today, she will be presenting her work that has been published recently in Nature Communication, titled Control of an epileptogenic circuit by the cannabinoid CB1 receptor-interacting protein GAP43. So Irene, thank you very much for being here with us. Thank you for your willingness to share your work with us. And please go ahead and thank you.
Thank you, Miguel, for the introduction. Let's see. Yes. So yeah, I come from Manuel Guzmán's lab, and our lab is focused now on new proteins that can interact with the cannabinoid CB1 receptor, shaping its function. And today I will talk about one of them, which is called GAP43. And it's involved in an epileptogenic hippocampal circuit. So a CB1 receptor is a G protein coupled receptor that belongs to this cell communication system called the endocannabinoid system. And together with its homologous receptor, CB2 receptor, are the best characterized cannabinoid receptors up until now. CB1 receptor is widely expressed in the nervous system and also in some other tissue, while CB2 shows a more restricted expression, mainly to cells of the immune system. These receptors are activated by endogenous lipid ligands, the endocannabinoids, such as 2-AG or anandamide, and also by its exogenous analogs, the phytocannabinoids derived from the plant cannabis sativa, such as the THC, the main psychoactive component of the plant. Apart from receptors and ligands, synthesis and degradation enzymes for these endocannabinoids are also part of this endocannabinoid system. So CB1 receptor is mainly located on the presynaptic neuron, where it plays a neuromodulatory role. So when the postsynaptic neuron is depolarized, endocannabinoids are synthesized and released, acting as retrograde messengers that travels back to activate CB1 receptor in the presynaptic terminal. The active receptor can then, by different mechanisms, inhibit neurotransmitter release, and in this way modulate synaptic efficacy in a short-term manner, including what we know as depolarization-induced suppression of excitation or inhibition, or in a long-term manner, what we know as long-term depression or LTD. Within the brain, high levels of CB1 receptors, shown here in purple, are found in the olfactory bulb, neocortex, the basal ganglia, the hippocampus, or the cerebellum, regulating, therefore, a large number of brain functions, such as cognition, food intake, motor control, anxiety, memory, or emotions. The pharmacology of this receptor is diverse, showing complex, even sometimes biphasic fine-tuning of some of these functions. For example, food intake, or anxiety, depending on the context where this receptor is expressed, leading to very different behavioral outcomes. And this complexity can be explained by the different signaling events that the receptor can trigger. Because CB1 receptor can activate several different signaling cascades. Canonically, it binds to GI protein and inhibits cyclic IMP formation, PKA, or calcium currents, or it activates potassium channels, or MAP kinases, or mTORC1 signaling cascades. But also, the receptor can couple to different non-GI proteins and therefore trigger different signaling cascades. And so the preference of one signaling cascade over another, this is the biased signaling mediated by the CB1 receptor, is dependent on contextual factors, such as, for example, the ligand for the receptor, endogenous or exogenous, the doses of these ligands, or the specific cellular environment of different cell types or neuronal populations. And this, in turn, triggers and alters synaptic plasticity or different patterns of neuronal activity. Another way to modulate this biased CB1 receptor signaling is conceivably by its association to certain intracellular proteins by its large cytoplasmic C-terminal domain. For example, some intracellular proteins have been described previously in the literature, such as the well-known CRIP1A, or some others such as FAN or GAS1, etc. However, none of these protein interactions with the receptor has addressed a physiological relevance of that interaction. So that's why in our lab we found interesting to look for still unknown neuronal type specific proteins that can directly interact with the receptor, and if so, to determine if this interaction can modulate the receptor function in a specific physiological or pathological context. So to identify new interacting CB1 receptor interacting proteins, we first express and purified the C-terminal domain of the receptor bound to a lectin, which allow us to anchor it to a Cepheros chromatography column, exposing the C-terminal domain. Then this column was challenged to a whole brain C-brain homogenate by affinity purification, and specifically bounded brain proteins were then eluted, digested, and the resultant peptides were identified by tandem mass spectrometry. So out of the list of hits that we obtained, we choose to focus our work on the protein GAFR3, also known as neuromodulin, due to its statistical significance and its role in the nervous system. And this is the protein that I will talk about today, and a story that we have recently published. We are very happy for that, but also we choose another protein from this list, cereblon, that some of my colleagues are working on, and that will be our next story coming soon. Okay, so GAF43 is a cytoskeletal associated presynaptic protein highly expressed in the nervous system. It shows an N-terminal domain that serves as membrane-targeted domain by reversible palmitoylation. and it is activated mainly by conventional PKC phosphorylation and a specific residue, serine, in position 41 within its effector domain. So that phosphorylated GAP then can interact with directly with actin or with other cytoskeletal or vesicular associated proteins to fulfill its functions in the presynaptic terminal. During development, GAP43 is a key factor in neurid outgrowth and axonal pathfinding. And in adult brain, it's involved in axonal regeneration upon axotomy, recapitulating development, and also in vesicle recycling, in long-term synaptic plasticity in hippocampus, and in learning and spatial memory performance. So upon this starting proteomic analysis, our aim was to characterize GAP43 as a CB1 receptor interacting protein, answering the following questions. Do they really interact in the brain? Where does it happen? Which type of terminal? And does it affect the receptor function on the brain, on plasticity or on mice behavior? So first of all, we focus on the interaction, and to evaluate a direct protein-protein interaction, first we purified a fluorophore-bound C-terminal domain of the receptor and the GAP43 protein to conduct fluorescence polarization banding assays. So briefly, when a fluorophore-labeled peptide is excited with polarized light, it will emit highly depolarized light due to the fast rotation of this molecule in solution. But if this molecule can interact with a high molecular weight protein, the rotational speed of the complex will decrease and the emitted light will remain highly polarized. So in our experiment, when we excite with polarized light a solution with labeled C-terminal domain and increasing concentrations of GAP43, we observed this saturating curve of increasing fluorescence, indicating the formation of a complex and therefore a direct interaction between both proteins in solution. But do they interact in a biological substrate? So we showed that endogenous GAP43 and CB1 are interacting by performing co-immunoprecipitation assays in primary mouse hippocampal neurons and dissected mouse hippocampal tissue, as we saw here with these arrowheads. But moreover, since both proteins are expressed in the presynaptic terminal, we isolated these terminals in synaptosomes preparations, and we conduct proximity ligation assays. This technique, this PLA technique, allows to detect interaction or close proximity between proteins by using primary antibodies against those specific proteins and secondary antibodies bound to DNA primers. So if both proteins are close enough, these DNA primers can hybride, and they can also undergo an amplification reaction with fluorescent probes that emit red light, so we can detect the complex. So as you can see in this image, we could see a PLA positive red signal in synaptosomes of hippocampus of wild-type mice that was drastically reduced in the synaptosomes of CB1 receptor knockout mice. So with these experiments, we could conclude that CB1 receptor and GAP43 were interacting in hippocampal terminals, neuronal terminals. Okay, next we wanted to know if, as we have seen in the literature for other proteins, GAP43 needs to be phosphorylated to interact with the receptor. So to modify the activation state of GAP43, we designed two mutant versions of the protein. One with a point mutation of the serine 41 by an alanine, creating a phosphorylation deficient mutant, so an inactive form of the protein. And another one with a point mutation by an aspartate, mimicking a constitutive phosphorylation of the of the protein, so making the active form, constitutively active form of the protein. And using these two tools, we conducted again PLA, now in hex cells, expressing the CB1 receptor and GAP43 wild type and the two mutant versions. And as you can see here, only cells expressing the GAP43 wild type form and the constitutive phosphorylated or active form showed evident PLA signal and significantly higher than the one we observed when the cells express the inactive form of GAP. Then we did two more protein-protein interaction techniques, such as co-immunoprecipitation and BREAD. And by these three techniques, we confirmed that GAP43 needs or requires to be phosphorylated to interact with the receptor in hex cells. And in this same cell line, we wanted to have a first insight on the impact of the complex on the receptor function. And so to address this issue, we made dynamic mass redistribution experiments, which evaluate changes in the global signaling of the receptor upon agonist treatment. Basically, when we add the ligand for a receptor, the active receptor triggers then all its signaling cascades, and this triggers a small cell mass redistribution that we can measure by or detect by change in the wavelength of the monochromatic reflected light. Basically, this is how this technique works. So when hex cells expressing CB1 receptor and a control vector are treated with the CB1 receptor agonist Win, we could register a response in the cell. Co-expression of CB1 receptor wild type or the active form of GAP partially block this Win-mediated response of the cell, which does not happen when we express the inactive form of GAP43. So this suggests that GAP43 is inhibiting CB1 receptor functions in the cell. Well, so once that we prove an interaction and maybe an inhibitory role of GAP on CB1 receptor functions, we aim at mapping this complex on the brain. And for that, we did immunofluorescence assays in different brain areas, and we saw that GAP43 and CB1 receptor are both expressed in striatum, cortex, or hippocampus, but the dentate gyrus of the hippocampal formation kept our attention, since both proteins were highly expressed in a very specific and restricted area of the dentate, the inner molecular layer, or IML. And this area is known for expressing high levels of CB1 receptor on glutamatergic terminals, as was shown in this work of Cristina Monori, published in 2006, where they used conditional knockout mice for glutamatergic neuron CB1 receptor or GABAergic neuron CB1 receptor. So we used these same animal models, and we did again our immunofluorescence experiments, and we could see that in the IML of the GABAergic neuron CB1 receptor knockout mice, there were double positive dots for CB1 receptor and GAB43 that were absent in the IML of glutamatergic neuron CB1 receptor knockout mice. So this indicates that, or suggests that GAB43 and CB1 receptor co-localize in glutamatergic terminals of the IML. But to test interaction between both proteins in this area, we conduct again PLA, and as you can see here, we could detect red dots corresponding to PLA signal in the IML of wild-type mice or GABAergic neuron CB1 receptor knockout mice, but they were drastically reduced in the IML of glutamatergic neuron CB1 receptor knockout mice or full knockouts for CB1 receptor. Moreover, here we wanted to try a rescue strategy in a null CB1 receptor background. So we saw that PLA signal was restored in the IML of mice where we rescue the expression of CB1 receptor in every cell of the mice, or specifically in glutamatergic neurons. So this reveals that glutamatergic neuron CB1 receptor plays a sufficient role for interact with CB1 receptor, with GAB43, sorry. Good. So, but what is the function of these receptors, these glutamatergic neuron CB1 receptors? Glutamatergic terminals of the IML mainly belongs to mainly corresponds to the MOSI cell axons that impinging here on proximal dendrites of granule cells, the principal cell type of the dentate gyrus, and granule cells project back to MOSI cells, establishing this recurrent excitatory circuit in the dentate that is important for consolidation of associative memories, but also for hyperexcitability-driven seizures. So then here in this synapse, MOSI cell to granular cell, CB1 receptors are highly expressed and mediate short-term forms of plasticity such as DSE or Wnt-mediated suppression of neurotransmission. And this was described firstly by the Pablo Castillo laboratory in 2008. So we wanted to know if CB1 receptor-mediated plasticity in this synapse was affected by the interaction with GAB43. So to address this question, I did stay in Pablo Castillo laboratory in New York, and as we know that GAB43 needs to be phosphorylated to interact with the receptor, we first produced some adeno-associated viral vectors expressing the two mutant forms of GAB43, active and inactive, under the control of a constitutive promoter and fused to a fluorescent reporter, CFP, or some CFP vector, and then we injected these viral vectors ipsilaterally in the hilus of 3-week-old mice. Then 3-4 weeks later, to allow viral expression, we made electrochemical recordings in the contralateral dentate, because we know that MOSI cells project contralaterally. So in this way, we can restrict our viral infection only to presynaptic terminals. As you can see here in these images of contralateral dentates, CFB expression is restricted to fibers in the IML, and it's absent from granule cells or the postsynaptic cell layer. So once we have our injected mice, we did coronal brain slices, and we simulate here the afferences of the MOSI cells in the inner molecular layer, and we place a recording patch pipette like 100 micrometers away to record the field excitatory postsynaptic potentials. This is the postsynaptic excitatory potential of the whole granular cell population. So in this recording setting, we could see that upon a 25-win treatment, we observe the expected reduction of the field EPSP amplitude only in mice injected with control vectors or with inactive GAP43. However, the Win-mediated suppression of neurotransmission was reduced in mice expressing the active form of GAP43. Similarly, in these same slices, we did a whole cell batch clone recordings. We, same as before, stimulate here the MOSI cell axons in the IML, and we recorded the excitatory postsynaptic currents of a sealed granule cell. So every time that we stimulate, we record EPSC, and once we have a stable baseline of EPSCs, we trigger a we injected a depolarizing voltage step inside the pipette to trigger the intrinsic release of endocannabinoids from the granule cell to the presynaptic terminals. So once we do this, we observe this transient decrease in the EPSC's amplitude that lasts less than 2 minutes. So in our experiment, we observed this canonical DSE in control-injected mice and in mice expressing the inactive form of GAP. Meanwhile, the DSE was blunted in mice expressing the active form of GAP43. So these two experiments suggest that phosphorylated GAP43, by interacting with CB1 receptor, is inhibiting C1 receptor function in this MOSI cell to granular cell synapse. At this point, in contrast with an overexpression strategy, we wanted to modulate the endogenous GAP43 of this synapse. So for that, we generated a GAP43 fluxed mice, and we injected these mice, or alternatively wild-type littermates, with a mix of two types of adeno-associated virus. One expressed CRE recombinase under the control of CAM kinase 2 promoter. This is for promoter specific for projecting neurons, fused to a M-cherry reporter, and another one that expresses an enhanced channel rhodopsin with a CRE-dependent expression, and also fused to a red TB tomato reporter. The channel rhodopsin is light-gated ion channel that activates the neuron where it is present. So when CRE is expressed in projecting neurons, it will recombine log-SPE sequences to delete GAP43, and in those same cells, it will allow the expression of the chief. And we confirm this in tissue by the absence of double positive puncta for GAP43 in green and T tomato red. Okay, so in this conditional knockout GAP43 mouse, we could see that Wnt-mediated suppression of neurotransmission. Wait. Hola? Sorry? Oh, okay.
Yeah,
so that
you're fine. Yeah.
Okay. So that the Wnt-mediated suppression of neurotransmission was much faster in this conditional knockout for GAP43 in mouse cells than in the wild-type littermates, so that the endogenous loss of GAP43 is in this case enhancing CB1 receptor functions. Okay, so now that we know that CB1 receptor GAP43 are located in this specific excitatory circuit, and that GAP inhibits CB1 receptor synaptic activity here, we aim at studying a possible impact on behavior. So for that, we crossed these flux GAP43 mice with mice that express the Cre recombinase under the control of a specific telencephalic glutamatergic promoter, next one, or with mice that express the Cre under the control of a telencephalic forebrain GABAergic promoter, DLX56, so that we generate two new colonies of conditional knockout mouse for GAP43, specifically on glutamatergic or GABAergic neurons. So both models were viable and fertile, and so reduced levels of GAP43 in the hippocampus, and specifically for the glutamatergic GAP43 knockout in the IML. So we decided to focus our study on epileptic seizures, because we knew that the dysregulation of this mouse cell to granule cell recurrent loop is a hallmark in temporal lobe epilepsy models. Also, it was described that the deletion of glutamatergic CB1 receptor in the dentate, mostly from mouse cells, worsens seizure severity. And third, because GAP43 has been proposed as a promoting factor of epileptogenesis. So with all this background, we designed the following experiment. We injected glutamatergic neuron or GABAergic neuron GAP43 knockout mice or their wild-type littermates with vehicle or THC 10 milligrams kilo, and 15 minutes later we injected them with the proconvulsant drug cyanic acid at 30 milligrams kilo to generate this well-established model of temporal lobe epilepsy in mice. And we recorded mouse behavior for the next 2 hours before euthanize them. And we evaluate their behavior according to the known modified Racine scale, which defines 7 behavioral stages of increasing seizure severity. So at this high concentration of kinates, in wild-type littermates, we could not see any difference in the progression of seizures over time between vehicle-treated or THC-treated mice. However, in THC treatment prevented the progression to more severe seizures in glutamatergic neuron GAF43 knockout mice, and also decrease seizure severity and conversely increase latency to develop seizures, compared to vehicle-treated glutamatergic neuron GAF43 knockout mice. And well, in contrast, THC had no effect on the GABAergic neuron CB1 receptor knockout mice. Finally, to support this behavioral data, we monitored electroencephalographic activity of the dentate gyrus in kinete-injected mice in the same experimental setting as before. And here we confirm that THC treated glutamatergic GAF43 knockout mice showed an increased latency to tonic seizure onset and decreased interictal spike frequency compared to vehicle-treated glutamatergic GAF43 knockout mice or their wild-type litter mates. So these results led us to conclude that that upon deletion of GAF43 on telencephalic glutamatergic neurons, this inhibited CB1 receptor would prevent from an epileptic phenotype triggered by kainate injection. And now then, to sum up all the study, in mosaic cell to granular cell synapse, when this granular cell is depolarized, endocannabinoids are synthesized and released to activate CB1 receptor in mosaic cell, so it can reduce neurotransmitter release and trigger DSC. Within this scenario, our data lead us to conclude the existence of a CB1 receptor interacting protein, GAF43, in mosaic cell that, upon phosphorylation, can interact with the C-terminal domain of the receptor and inhibit its function. And by electrophysiological techniques, we could discover that phosphorylated GAP reduced short-term forms of plasticity mediated by the receptors, such as DSC and Wnt suppression of neurotransmission. And conversely, GAF43 deletion from mosaic cell enhanced synaptic activity of the receptor. And this will lead to an enhanced CB1 receptor-mediated anti-seizure activity in this circuit. Therefore, GAF43 could be a distinctive molecular factor that helps in the mechanistical understanding of the role of this receptor in a recurrent excitatory circuit and its involvement in the control of epileptic seizures. Thank you very much to all my group that made this possible, and all the collaborators, and all of you from CIBERNED for listening today. Thank you.
Thank you very much, Irene, for a very nice presentation. So the paper is now open for discussion. So as I said earlier, if anyone is interested in making a question, please do so by raising your hand using the icon in the option in the Teams application, or you can also send me a message through the chat, and I will pass the questions to Irene. So while we wait for the first questions, let me just remind everyone that CIBERNED Webinar Series are held every other weeks on Thursdays at 3:30 p.m. And we have now sent an email to everyone communicating that the call for next semester webinars is now open. So if anyone is interested in participating in this webinar series, please do so by sending a email message to Teresa Iglesias, which is the training coordinator at CIBERNED, and stating that you are interested in presenting in this series. Okay, so I don't see any raised hand yet, or any messages coming through the chat. But again, while we wait for questions, let me also make an advertisement. So we'll have a special webinar next week on Thursday. It will be a table round format to discuss the recent approved treatments for Alzheimer's disease, in particular the lecanemab monoclonal antibody. So it's similar webinar as we did a year and a half ago, if maybe some of you remember, when we discussed the first approval of the other monoclonal antibody, the aducanumab. So this will be on Thursdays, as I say. It will be in Spanish, and we will have a roundtable format for it. Okay, so I think we have we have a first question coming from Teresa Iglesias. Teresa.
Hello, do you see me? Yes.
Yes, we do.
I'm here. So thanks very much for your talk. It's a huge work. So congratulations as well for the publication. I think it's a great work. I don't know, maybe you said it and I got lost in the way, but so you see that the use of cannabinoids for inhibiting epilepsy, the mechanism by which this is doing so is, do you think is inhibiting the release of glutamate in the presynaptic terminal, or what is your? Probably you mentioned it and I didn't notice, but could you explain that?
Yes, here in this particular circuit, and in this case, yes, I think it would be the receptor inhibiting the release of excessive glutamate, because in this case, MOSI cells in an epileptic context, they become very active. So they should be releasing a lot of glutamate. So here, probably CB1 receptors are reducing that excess of glutamate. Yes.
For the therapeutic applications, probably you discuss this in the paper, or you have any? Because the circuit is really nice described, and you dissect very well the molecular mechanism by doing all these, mice and mutants. But do you think it would have clinical applications for anti-epileptic treatments?
Yeah.
We should not go to that. I mean, I love basic knowledge and that, but normally they always force us to speculate at least.
Yeah. So we should be like cautious when we speak about THC treating epilepsy, because THC has also like psychoactive connotations, and that's why now it's epilepsy is mostly treated with or in clinic with CBD. But yeah, this is basic science, but yeah, in the future would be maybe nice to try to break this interaction with GAL43, maybe, if also we don't know if this interaction is enhanced in an epileptic context, but maybe if it is, if we break the interaction with GAP specifically somehow in that circuit, then we could apply this to clinic, maybe. But
because if I'm not confused, so for the effect you need it to be phosphorylated by PKC, right? Have you tried to inhibit PKC and see what happens?
Yeah, no, but that would be good to inhibit PKC specifically there, and see if that's the endogenous mechanism for the activation of GAP in these mossy cells. Yes. Yes.
Oh, lovely work and a great presentation, Irene. Congratulations.
Thank you.
Okay, thanks.
All right, thanks, Teresa. And so, any more questions? I don't see any other raised hand. Nor I see any message coming through the chat. Okay, so this is the last call for questions to Irene. This is your last chance.
Well, yeah. They can
More
ask me
questions?
later also if something comes up.
Sure. Okay,
by
so
email.
yeah, so if there are no more questions, then I think it's time to close the session here. But no, before thanking Irene for her willingness to share with us her work, and congratulating her for the paper, and thanking everyone for attending. And I hope to see you in the next webinars. Thank you, Irene. Thank you all. Have a good day.
Thank you. You too.
Bye-bye.
Bye.